Okay. Hi, my name is Ayella-International. I am a PhD student at Virginia Tech, and I'm going to be talking about antibiotic resistance upstream and downstream of wastewater treatment plants at multiple international locations. So first, I'm going to go over some of these key concepts. Antibiotic resistance is a natural phenomena, so bacteria can have genes that allow them to survive in the presence of antibiotics. However, with the high use of antibiotics in clinical and agricultural settings, we've seen an increase in resistance genes in bacteria. And so we are in particular interested in genes that allow bacteria to survive in the presence of antibiotics that are used in clinical settings, and especially antibiotics of last resort. The reason that we're interested in water and the environment is kind of twofold generally. So the first being that there can be transfer of genetic material between the environmental bacteria and pathogens. And so we don't want there to be an environmental bacteria that has a resistance gene that it can then pass to a pathogen, and then you have a pathogen that's resistant to another antibiotic. And the other reason is just making sure that waters are safe or as safe as possible for recreational use. The other main component of our study are wastewater treatment plants. And the reason that we look at these is because they can be potential hotspots for disseminating antibiotic resistance. And the reasons are because when people consume antibiotics, some of those antibiotics or their metabolites are actually excreted and make their way to the wastewater treatment plant. Some wastewater treatment plants also receive industrial waste, and these can have components that co-select for antibiotic resistance. Within the wastewater treatment plant process, one of the steps involves a high density of microbes that are degrading the organic matter in the waste. And so you can have, again, exchanging of genetic material while there's these high density of microbes, and if there's selective pressure, you could then be increasing the bacteria that have antibiotic resistance genes. And of course, wastewater treatment plants discharge their treated water to the environment, so they're a place to monitor. So in terms of the literature, what we've seen in the past, people have done studies looking at antibiotic resistance genes, antibiotic resistant bacteria, as well as antibiotics, both upstream and downstream of wastewater treatment plants. And so we went into our study with a similar hypothesis that we would see an increase in antibiotic resistance genes downstream of wastewater treatment plants in all of our international sampling locations. So our study was a big collaborative effort. You can see our collaborators up in the corner there. And we sampled multiple wastewater treatment plants at each of the different stages throughout the treatment process. This study specifically that I'm going to be presenting is focusing on the receiving environment, so as I said, upstream and downstream. And we'll be looking at Switzerland, Sweden, the US, and India. And we analyzed our samples via Qpcr and Metagenomics. So very briefly, Qpcr is a rather fast method. It allows you to be very specific, so you can target a certain gene, and you have pretty high sensitivity from those results. Metagenomics, on the other hand, is sequencing all of the DNA that you find in your sample, so it's an unbiased approach because you don't have a target. It has a higher throughput, so you can run many samples all at once, and this expanded discovery comes in because you don't have a target, so you can look at all of the different DNA fragments that were in your sample. So first I'm going to be talking about our Qpcr results. So these are going to be normalized to our 16S rRNA gene, and we looked at 5 different antibiotic resistance genes as well as an integrase 1 gene. And I'm going to be showing a box plot that shows upstream samples in blue and downstream in kind of a green-brown color. So here you can see that there does appear to be a trend downstream of wastewater treatment plants. There seems to be this increase in antibiotic resistance genes. And we see this for all of our genes grouped together. And here we've pulled out SOL1, which is a sulfonamide resistance gene. So we can see that the trend appears to match with what other literature has found. So next I'm going to be showing some of our metagenomic data, and it's going to be in the same kind of format. So here we have, you can see many, many more genes. These are all of the antibiotic resistance genes that we detected in our samples. And we can also see that the pattern changes. You can no longer see a clear increase downstream of the wastewater treatment plant. And this is because we've kind of stepped back and we're looking at a more aggregated view of all of our data. So with metagenomics, we can look really specifically at certain genes or certain changes. And as I mentioned earlier, we're interested in clinically relevant antibiotic resistance genes. So I've highlighted them here in red, and you can see some locations have clinically relevant antibiotic resistance genes or ARGs downstream but not upstream, and we can clearly see that certain locations have a very high number of clinically relevant genes. And the clinically relevant genes that are shown here are either resistant to beta-lactam, peptide, or quinolone antibiotics. So this next slide is showing that same data that I just showed, but broken down in a different way. So instead of putting all of our genes where they all look the same, we've now broken them down based on the antibiotic that they are resistant to. And so again, we're looking at upstream versus downstream. So here you can clearly see, similar to our previous graph, we have a lot more samples, or a lot more antibiotic resistance genes in our samples from India. And again, there's not a clear difference or a clear increase downstream of the wastewater treatment plants. However, we can see that there are a few samples that have significantly different samples. And one of the things to note about these is just sampling locations. So the three that are significantly different were sampled closer together, whereas some of the other ones, there was a greater distance between the upstream and downstream samples, and they were further away from where the final effluent is discharged into the stream. And this is just based on, environmental sampling, where you can get to the stream and things like that. So this was a kind of brief overview of some of our analyses, but there are many, many more things that you can do with metagenomics data, looking at functionality and getting more in-depth into all of the genes that we found in our samples. But bringing this all back to our topic, what's next, emerging concerns. So the concern that we've discussed here is antibiotic resistance, and more specifically focusing in on genes that allow bacteria to survive in the presence of antibiotics used in clinical settings. In terms of what's next, the study that we did was possible because of all of the international collaborators that we had. So continuing these kinds of global collaborations is really important because antibiotic resistance knows no borders. Changes to policies are also really important. One that has been implemented in the past, some countries have restricted which antibiotics can be used in agricultural settings, and of course, monitoring other targets. So the methods that I've shown here can be used to look for other things, not just antibiotic resistance genes. Very relevant to right now, our lab actually does something similar to look for COVID in wastewater. So with these kinds of methods, you can look for a particular gene with Qpcr. So for example, looking for a gene that correlates to COVID infections, but you can also use metagenomics to look for novel genes that you might not have expected, or a more aggregated view of your samples. Thank you.